So now let's take a look at DNA replication itself, how this process works. When we look at the grand scheme of things, we only really require three things. First of all, something that we need to copy, and that would be the parental DNA molecule. Something to do the copying. There's a number of different enzymes involved in the copying. And then building blocks to make the copy. So we need nucleotides to put in a string to duplicate the DNA molecule. These nucleotides come as triphosphates, so they have their own energy with them in order to fuel the formation of the phosphodiester bonds. There are three phases to DNA replication. The initiation, when we assemble all the enzymes in the right place to begin the whole process. The elongation, which is where the new strands of DNA are synthesized by enzymes called DNA polymerases. And then termination, where replication is terminated. On this figure, you can see the nucleoside triphosphates. As you'll recall, the next nucleotide in the strand is attached to the 3' OH group of the growing strand. This is important when we consider directionality of how the enzymes move. Let's take a quick look at some polymerases. There are two types of polymerase. Polymerase means polymer, ase means enzyme, so an enzyme that polymerizes, as in adds subunits to the strand. DNA polymerase is a polymerase that's going to match existing DNA bases with complementary nucleotides and link them together. The DNA polymerase, you can see, is over here, adding nucleotides to the 3' end of the growing strand. We have the template strand down here, or the parental strand, and then the growing strand up here. Nucleotides are added to the 3' OH group of each preceding nucleotide. We also see up here RNA polymerase. RNA polymerase is only involved in making the primer because DNA polymerase has to have a handle to attach its nucleotides to. So it's really important that we have an RNA polymerase in order to build the primer, which is made up of RNA nucleotides. Once the primer's there, DNA polymerase can attach nucleotides to the end of the primer and grow the DNA strand. Of course, later on, the primers need to be replaced with DNA nucleotides. We're going to run into several DNA polymerases: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. All of them have common features. One is that they add bases to the 3' end of existing strands, and they synthesize in the 5' to 3' direction. So the 5' 3' direction is that we are adding a base to the 3' OH, so it grows from 5' towards the 3' direction. So this new strand is growing towards the 3' direction because we're adding nucleotides to the 3' OH group. So we say that DNA is synthesized in the 5' to 3' direction. I think of it this way. DNA polymerase can only read 3 to 5. So it's reading from 3' to 5' on the template strand and laying down new nucleotides in the 5' to 3' direction on the growing strand. So DNA polymerase can only read the template strand from 3 to 5. Thus, the complementary strand is going to be laid down from 5 to 3. And again, the importance of a primer. All DNA polymerases require a primer of RNA. It's sort of like thinking about painting a room. We need primer so that the paint can stick to the primer. It's much the same here. We need an RNA primer so that the DNA can stick to it in order to grow the growing strand. Now, most of what we understand about DNA replication was discovered in the E. coli, bacterial DNA replication. Keep in mind that bacteria have a single circular molecule of DNA. We'll note that replication begins at one origin of replication and proceeds in both directions around the chromosome. And we call the DNA controlled by an origin a replicon. In this case, it's the chromosome plus the origin forming a single replicon. And although we'll be primarily looking at DNA replication in prokaryotes, keep in mind that the process is very similar in eukaryotes. We'll look at some differences later on. E. coli will have 3 DNA polymerases. Recently, researchers have discovered several other polymerases, however, none of them are involved in DNA replication. The 3 polymerases are named in order of their discovery: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. DNA polymerase 3 is the main replication enzyme. It's the one that does the majority of elongation of DNA strands. DNA polymerase 2 is involved in repair, and DNA polymerase 1 is the polymerase that removes primers and replaces the RNA nucleotides of the primer with DNA nucleotides. All of them have 3' to 5' exonuclease activity. That is, they can proofread. An exonuclease is an enzyme that can break the phosphodiester bonds on the ends of the DNA as it's being replicated. The analogy I have here is it's as if there's a little van. DNA polymerase is a van driving down the strand, driving down the template strand in the 3' to 5' direction. There's a guy in the front seat, and he reads the template strand and says, "I see a T," and he tells the guy in the back sliding door to give him an A to pair with that. And then there's a guy that's sitting on the tailgate that's flopped down, and that guy is proofreading. And if he sees that there's a mispairing, like a C got thrown out instead of a T to pair with the A, then he asks the driver to back up, and they pull out the bad nucleotide and put down the right nucleotide. So this is 3' to 5' exonuclease activity. It's like backing up along the new strand. So the exonuclease is working on the newly formed growing strand. 3' to 5', we're just backing up along it a moment in order to pull out the wrong nucleotide and put the new nucleotide in. Now, DNA polymerase 1 has 5' to 3' exonuclease activity. That is, it can replace nucleotides in the forward direction. There's no backing up necessary. That's how DNA polymerase 1 has the ability to replace the primers that are laid down in order to begin growing the growing strand. The process of DNA replication requires that we unwind the DNA strand. Helicase is the enzyme that's used to unwind the DNA helix. I imagine it like a little wedge that goes in between these strands and pushes the DNA strands apart. In order for all the enzymes to access the DNA and actually be able to read it, we need to hold the replication fork open. This is held open with single-stranded binding proteins, or SSBs. They coat the strands and keep them held apart so they can't bind back to each other again. As we unwind the DNA and put enzymes in there to replicate the DNA strand, we'll see supercoiling of the DNA. Much like you see in a phone cord or a blow dryer, it gets all twisted up on itself. Topoisomerases are enzymes that prevent supercoiling, and the specific topoisomerase that we see in DNA replication is called DNA gyrase. It essentially cuts the DNA strand, untwists it, and re-glues the base pairs back together in order to prevent this supercoiling. Now, because DNA polymerase can only synthesize in one direction, remember it can only read the template strand, which is the green one in this case, from the 3' to the 5' direction, which is okay for one strand. We can lay down the primer at the beginning and lay down new nucleotides, attaching them to the 3' OH group and growing this strand. We call this the leading strand because its synthesis is continuous. As the replication fork opens up even more, we can continue along in the same direction. However, as the replication fork opens, the other strand is only presented in a 5' to 3' direction, and DNA polymerase cannot read in that direction. So this is the discontinuous side. As the replication fork opens, a primer's laid down, and we synthesize DNA nucleotides to the end. That leaves us one chunk. As the fork opens some more, we have to jump back and begin again. So we need another primer and then some more DNA nucleotides. As it opens up some more, we jump back, lay down a primer, add nucleotides. And this continues all the way along this other strand. We call the other strand the lagging strand because it has to keep jumping back, and thus it lags behind a bit. So the leading strand has continuous synthesis, and the lagging strand has discontinuous synthesis. This leaves us with fragments of DNA broken by primers. These are called Okazaki fragments. Later, these fragments will be glued together after we remove the primers with DNA polymerase I. So again, each Okazaki fragment has an RNA primer followed by DNA nucleotides. Later, each of these primers need to be removed by DNA polymerase I, so these RNA nucleotides can be replaced with DNA nucleotides. So the leading strand involves a single priming event. We have one primer and many primers on the lagging strand. The strand is extended by DNA polymerase III. So DNA polymerase III is the one that lays down each of these different nucleotides. You can see DNA polymerase III up here. DNA polymerase III is said to have a high degree of processivity. Processivity is simply the ability of the polymerase to remain attached to the DNA strand. It has this high degree of processivity because a beta subunit forms a sliding clamp. So we have DNA polymerase and it encircles the whole DNA strand, and thus it's called the sliding clamp model. This helps it stay attached to the DNA. We'll see how this works shortly. The lagging strand has discontinuous synthesis. DNA polymerase III, again, is the polymerase that elongates each Okazaki fragment as it attaches nucleotides to the primer that's been laid down by RNA polymerase. Each of the primers are laid down by an enzyme called primase. Once DNA polymerase III has synthesized its fragment of DNA and reached the beginning of the preceding primer, it comes off the strand, and the primers are removed and replaced by DNA polymerase I. Then what we're left with is a series of fragments that are all DNA, but they're not stuck together. So we need another enzyme called DNA ligase in order to glue the strands together. I think of DNA ligase as a little tube of glue. Termination on the leading strand will occur at a specific site. DNA gyrase will unlink the two copies and we'll have two whole new molecules of DNA. So as you're probably beginning to understand, there are a number of different enzymes involved in DNA replication, and you need to know what each of them are. They each have a specific role, and it's integral to the replication of DNA. Most of these enzymes are held together in a macromolecular assembly that you can see down below. We call this the replisome. There are two main components of a replisome. There's the primosome, which is made up of primase, helicase, and some accessory proteins right here. And then this portion of the replisome, which is a complex of two DNA polymerase IIIs, one for each strand, and they're held together by this clamp loading apparatus. A DNA polymerase III enzyme is active on each strand. Then primase synthesizes new primers for the lagging strand. There's a loop in the lagging strand template, which allows the replication to occur in this 5' to 3' direction on both strands because the polymerase can only read from 3' to 5'. The growing strand grows in 5' to 3' direction because it can only be read on the template strand from 3 to 5. Thus, new nucleotides on the complementary strand will be laid down from 5 to 3. When the polymerase III on the lagging strand hits the previously synthesized fragment, it releases the beta clamp and the template strand so that DNA polymerase I can replace the primers with DNA nucleotides. Now, A picture's worth 1,000 words, but a moving picture is worth way more. So let's look at this process as an animation.
The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
So naturally, we'll be diagramming this process. A really good place to start is by naming all of the different enzymes involved in DNA replication. So let's begin by listing all of our enzymes. What's the first thing that happens? DNA needs to be unwound by the enzyme helicase. And which enzyme holds the strands wide? Single-stranded binding proteins. And in order to stop the DNA supercoiling, we have the enzyme gyrase. In order to actually begin DNA replication, we have to lay down primers with the enzyme called primase. And following primase, we'll have DNA polymerase III. Primers will then be replaced by DNA polymerase I. The Okazaki fragments will be joined together by ligase. And I think that about covers it. So now start building your diagram by putting all of these together in sequence. It may look like the figure in the book, or it may be something completely different. You don't have to draw them all in place in the replisome. You can draw them separately if you like. Now let's take a quick card quiz to finish off this section. Enzyme that synthesized the new DNA strand during replication. What is the function of the gyrase enzyme? Where are the Okazaki fragments found? Name the enzyme that links the Okazaki fragments together. During DNA replication, which enzyme is responsible for untwisting the DNA helix? Here are your answers. If you have all of those, you're well on your way to understanding replication. It's a complex process, but really cool once you understand it.